Hello to everyone. Today we will discuss about the plant tissue. Firstly, we will discuss about the tissue. Tissue is a group of similar or dissimilar cells having common origin and responsible for performing similar function. Tissue are categorized into two groups, that is meristematic tissue and permanent tissue. Meristematic tissue. Meristematic tissue is composed of immature young cells capable of repeated division. The meristematic cells are thin walled, spherical, oval, or polygonal in shape, densely filled with protoplasm, having large nuclei and are actively metabolizing. The cells have the capacity of division. They grow and form the plant body other than embryonic cells. Meristematic tissue are of different types based on different sources. On the basis of origin, it is of two types, that is primary and secondary meristematic tissue. If we will discuss about the primary meristematic tissue present from beginning and persist throughout life, responsible for primary growth, lie mainly in the apices of stem and roots, hence as to length of root and shoots. Secondary meristematic tissue arise from the non-meristematic tissue by differentiation of permanent cells at later stages in life. For example, cork cambium and interfascicular cambium. On the basis of location, it is of three types, that is apical, lateral and intercalary. Firstly, we will discuss about the apical meristematic tissue. Apical primary meristem located in growing points, that is root and shoot apex, responsible for primary growth, result in increase in length. Next is lateral situated in older parts parallel to long axis of organ. These divide and responsible for increase in girth, that is secondary growth. For example, cork cambium and vascular cambium. नेक्स्ट है इंटरकैलरी, सॉरी यहाँ पे इंटरकैलरी होगा intercalary, located between region of permanent tissues, for example at nodes, and responsible for growth in length in region other than tip. On the basis of plane of division, it is of three types, मास मेरिस्टेम, प्लेट मेरिस्टेम, यहाँ पे प्लेट होगा, प्लेट मेरिस्टेम, रिब एंड फाइल मेरिस्टेम. Firstly, we will discuss about the mass meristem. Mass meristem cell division occur in all planes leading to increase in volume. For example, development of endosperm and embryo. Next is plate meristem. Plate meristem cell division occur only on two planes, showing increase in areas. For example, epidermis. Rib and file meristem cells divided only on one plane, increasing in length. For example, development of cortex and pith. Next is on the basis of function, it is of three types, that is protoderm, protocambium and ground meristem. So firstly, we will discuss about the protoderm. Protoderm, outermost meristematic layer of apical meristem which develop into epidermis luteum. Procambium located inside protoderm and give rise to primary vascular tissue. Ground meristem, major part of apical meristem which precursor of ground tissue such as pith, cortex, hypodermis, and pericycle. Next is root apex organization. Root apex consists of mass of meristematic cells. Due to presence of root cap, the root meristem becomes subterminal in position. There are theories which is given for root apex organization are histogram theory, which is histogram theory. According to this theory, the root apices consist of central core called plerome, surrounded by several layers of peribellum, outermost single layer of dermatozoon and calyptrozoon. Calyptrozoon give rise to root cap, dermatozoon to epidermal tissue system, and peribellum give rise to ground tissue system, and plerome give rise to vascular tissue system. नेक्स्ट है कॉर्पर कैप थ्योरी. कॉर्पर कैप थ्योरी given by Scoop 1917. According to this theory, the cell in root apex divide into two planes called T divisions. Quiescent central theory given by Claus. According to this theory, root apex consists of an inverted cup-like region of the quiescent center. The cells of this region have low amount of RNA, DNA, and protein and have low dividing activity. These are reverse meristem. Shoot apex organization. Shoot apex are present immediately above the youngest leaf primordia. It consists of meristematic cell. Theories are explained. Shoot apex organization are apical cell theory, histogram theory, tunica corpus theory. In apical cell theory, according to this theory, a single apical cell lead to development of entire plant body. Histogram theory. According to this theory, shoot apex consists of meristematic zone, the histozones, which are dermatozoons, that is forms epidermal tissue system, peribellum forms ground tissue system, and plerome form vascular tissue system. Next is tunica corpus theory. According to this theory, the shoot apex consists of tunica. Tunica means which form epidermis, and corpus means which form and central mass. Next is permanent tissue. These tissue are made up of cells that have lost of power of division and are differentiated to carry out various functions. The cells are living or dead. Permanent tissue are of three types, that is simple, complex, and spatial or secretory tissue. Firstly, we will discuss about the simple tissue. It is homogeneous in nature and composed of similar cells. It is of three types, that is parenchyma, collenchyma, and sclerenchyma. Firstly, we will discuss about the parenchyma. Parenchyma composed of living, isodiametric, spherical, oval, or polygonal cells with intracellular spaces and thin cellulosic cell wall. The main function is storage of food. The parenchymas storing some substances like tannins, resins, and gums. The parenchyma having large intercellular spaces are called arenchyma. The parenchyma having chloroplast called chlorenchyma, which help in photosynthesis. Next is collenchyma. Collenchyma consists of living cells, but cellulose and pectic substances are deposited at the corner of the cells, present in outer region of cortex of stem, leaves, petiole of dicot plant, and absent from root and monocote plant. It is mechanical tissue which provides support, flexibility, and elasticity to the organ. Next is sclerenchyma. Sclerenchyma consists of dead cell which thick lignified cell walls provide support and mechanical strength. It is consist of following two parts, that is sclerate, it is also called stone cells, and sclerenchymatous fiber. Firstly, we will discuss about the sclerate. Sclerate is stone cells lignified, extremely thick walled, so that the lumen of the cell is almost obliterated and may be spherical, oval, cylindrical, T-shaped, even satellite. Found in hard part of plant, for example, endocarp of walnut and coconut. And sclerenchymatous fiber, they are long and tapering at end. The cells are lignified and dead, commonly found in pericycle of stem, forming a solid tissue protecting the vascular bundle of dicots. Next is complex tissue. It is composed of more than one type of cells. First is xylem. It is two types, that is xylem and phloem. Firstly, we will discuss about the xylem. Xylem form the bulk of roots and stem, conducting water and minerals and provide support. It is composed of trachea, vessel, xylem, parenchyma, and xylem fiber. Firstly, we will discuss about the trachea. Trachea are elongated tube-like dead single cell with hard, thick, and lignified walls. End walls are tapered and overlap with adjacent trachea. Water passes trachea to trachea through the pit in their walls. Vessels. Vessels conducting unit of xylem, long tubular structure formed by end to end, consist of fusion of several cells. The first vessel form protoxylem. Lining is deposited in the cellulose wall of these vessels so as to form annular and spiral thickening and forming metaxylem. The long vessel transport large amount of water and minerals over long distances. Xylem parenchyma. Xylem parenchyma, living, thin-walled, abundant parenchymatous cells involved in storage of starch, sugar, and lipid. Xylem fiber. Sclerenchymatous occur abundantly woody dicots having thicker wall and narrower lumen, stronger and provide mechanical support. Next is phloem. Phloem, also called bast. It is called bast. Bast. Main function is transported of food. It is composed of sieve tube, that is sieve tube. Sieve tube, long, slender, tube-like structure involved in transport of organic solutes formed by end-to-end fusion of sieve tube elements. The wall of sieve tube element are made up of cellulose and pectic substances, but the nuclei degenerate at maturity. Two adjoining end wall of the neighboring sieve element form sieve plate remain alive by depending upon companion cells. So we will discuss about the companion cell. Companion cell, a thin-walled elongated cell, is associated with each sieve tube and are connected by simple pits. Companion cell is living, contain dense protoplasm and large elongated nucleus. Next is phloem parenchyma, which is composed of living and cylindrical cells. These are absent in most of monocot and mainly store food material. Phloem fiber, sclerenchymatous and provide mechanical support. Phloem fiber is jute, flax, and hemp are rotted in water and used for making rope and textiles. Next is special and secretory tissue. It is of two types, that is lentiferous tissue and glandular tissue. Glandular tissue are also two types. So first we will discuss about the lentiferous tissue. Lentiferous tissue made up of thin-walled, elongated, branched and multinucleate substances structures that contain colorless, milky or yellow colored juice called latex. Latex cell, they do not fuse, hence not form network. Plants having such tissue are called simple or non-articulated lentifer. It is example nerium and latex vessel formed due to fusion of cells and form network like structures. Example are papaver. And glandular tissue include different type of gland which secrete oil, gums, mucilage, tannin and resin. Thank you for watching. Thank you for watching.